The impact of excessive alcohol intake on vitamin C transporter expression levels in the brain. These transporters transport vitamin C into the cell through carrier-mediated processes, which transport vitamins into the appropriate compartments in the brain cells. The transporter expressions at the plasma membrane are the major route for vitamin uptake that are essential for a major spectrum of biochemical reactions, which lead to normal cellular function of brain cells. How does excessive alcohol intake impact vitamin C transporter expression levels of the sodium-dependent vitamin C transporter 1 and the sodium-dependent vitamin C transporter 2 in the brain? This process is a pathophysiological investigation which examines transporter gene expression levels transporting ascorbic acid by carrying out two experiments in hopes of emphasizing the damaging effect of the consumption of alcohol on the brain. That RNA was isolated from the brains, then primed with primers to synthesize first-strand DNA. The coding region of both the transporters were amplified. RT-PCR was then performed to normalize the data, which included in the internal control. It was then quantified using a relationship method supplied by the iCycler. Hypothesis 1 ended up not being supported because there was only a slight difference between control variable 1 and the sodium-dependent vitamin C transporter 1. The sodium-dependent vitamin C transporter 1 was only slightly affected, showing that hypothesis 1 was not supported due to just approaching statistical significance. Hypothesis 2 was supported by the experiment. When comparing control 2 and sodium-dependent vitamin transporter 2, there was a clear difference between the two groups. There was significant change between the gene expression levels transporting vitamin C and the corresponding experimental brain samples. In conclusion, vitamins can play a vital role in the treatment of brain disorders and for cognitive development of children and teens, proving the need to protect the sodium-dependent vitamin transporter 2 by not consuming alcohol.